Hello, we have a new update to this project. As a quick reminder, what we did was we segmented these cross sections of muscle fibers and we manually segmented them, and we excluded any shapes that were cut off by the border. These were the 4 groups that we analyzed: young control mice, young mice treated with amibegrin, you have aged controls, and aged mice treated with amibegrin. Important detail to know is that after we isolated each fiber particle, we oriented such that the longest internal length was vertical, and we did this because it cleans up the data. Because nature is inherently polar, and therefore when we align shapes according to those poles, we get cleaner data. Here's a reminder of how the LCPC transform works. I will not go into details. And this is a reminder that since this is a particle system, the way we compare it between different groups is we only compare the same quartile across different groups. And we calculate quartiles as an objective way of identifying natural subgroups. And we do this because treating the whole system in bulk drowns out a lot of interesting biological insights. All right, for some housekeeping, right, from last time we increased the sample size, right, and thus we call it batch 2. And I just want to show that batch 2 is comparable to what we found in batch 1. So here from batch 1, our young controls, we had 6 samples, but in our other groups we only had 3, and some of these were from the same animal. And so we wanted to increase the sample size for more distinct animals, and we did that for all these 3 groups. And as you can see here, just comparing the particles from every single group, that the data is comparable between the 2 batches. Other than this one sample here in the aged control mice, which seems a bit odd, but given the natural variation in biology, this is not surprising or concerning. Okay, and just for housekeeping, this is just to show you here we measured pure shape. All right, that is we resized the shapes according to their longest internal length. And here's the results in batch 2 and batch 1 at the bottom. As you can see that the patterns are comparable. Same for when we measure these particles at scale, that is at their original size and shape. The patterns between batch 1 and 2 and batch 1 are comparable. So now let's move on to interpreting the results. So what is the effect of amibegrin on fiber shape and fiber size? Well, let's start with quartile 1. And as a reminder, quartile 1 are the smallest cells, right, in terms of size, okay, in the particle system. And there's a minor difference or no difference between these in young mice, right? In blue is young controls and orange is a young amibegrin, and that there's no difference between size and shape measured together versus just pure size. But if you look at the aged group, it's very interesting is that when you measure size and shape together, amibegrin increases, right, the shape magnitude, while if you measure pure shape, amibegrin causes a decrease in the shape magnitude. And this does make sense because if you look at here shape and size, right, overall, if you just eyeball these images, right, the size of each fiber here is larger than the shape of the typical fiber in the amibegrin group, right? So that makes sense of this increase here. In terms of this decrease here, this makes sense too, the image does, that is, because if you look at the aged group, the edge of the fiber is very jagged, right? And we made sure to outline the jaggedness to be true to the morphology. And the effect of amibegrin is to smoothen out that edge. And so the smoother the edge, right, the less information is quantified, okay, or that results in a smaller shape magnitude, which is reflected when we measure pure shape. All right, and so next let's look at quartile 2. So now we've entered the mid-sized particle region, quartile 2. If you look at the young group, there's a difference in due to size and shape when we measure it together. But that difference disappears when we measure pure shape. So what this tells us is that the effect of amibegrin in young mice is to change the size more so than to change the shape. All right, and then in the aged group, measuring size and shape together shows a minor reduction, But when you measure pure shape, there's a major reduction. So what this tells you is that the effect of amibigrine in aged mice is distinct from young mice. That is, in aged mice, amibigrine causes a major change in shape, and less so in size. And we see here an inverse effect of amibigrine in terms of what it does in young mice and old mice, and this theme will persist in the next quartile that we will discuss. So quartile 3 now is still the mid-size one, the mid-size particles in the system, but slightly larger. And we see here that in young mice, amibigrine causes a decrease in size and shape together. But if you look at pure shape, it's the same, right? So the effect of amibigrine in young mice is to reduce the size of the fiber more so than the shape of the fiber. The opposite effect is present in aged mice. Here, if you measure size and shape together, there's not much change. But if you measure pure shape, there's a major change, right? So in aged mice, amibigrine changes the shape more so than it does the size of the fiber. Lastly, we reach quartile 4. These are the largest particles in the system. And if you measure in young mice size and shape together, you see a drastic reduction. When you measure pure shape, there is still a reduction, right, in shape. If you look at aged mice, if you measure size and shape together, you see a reduction. But when you measure pure shape, that reduction begins to disappear, right? And so the effect of amibigrine in aged mice versus young mice is distinct in terms of morphology. And here are the quartiles together, just for you to peruse. In terms of conclusions, you can read the details at the top here, but there's some redundancy, so I'll just highlight the bottom points. The amibigrine affects muscle shape more in aged mice than it does in young mice. The effect of amibigrine in young mice is primarily a size difference as opposed to a shape difference, while the effect of amibigrine on aged mice is primarily a shape difference as opposed to a size difference. And lastly, amibigrine reverses the shape of fibers in aged mice, but not to the levels of young mice. And the last few slides in the appendix are just enlarged graphs with descriptive statistics and p-values, whatnot. I will let you peruse that. So that is the update. Let me know if you have any questions, and I'd be interested to hear how this data matches with the gene expression data and other, phenotypic readouts that you found.